Glad to speak and give an update of our research. I presented some initial findings of our genomic research on dama gazelle genomics back in the 2018 meeting. And so will be an update. So I don't have to talk about the history of the dama gazelle because Teresa and Sonia and Rathak did a very nice job of already talking about and presenting what the perils that face this species. It is amazing that this species, 95% of the population is found in captive populations. And Teresa and Sonia's talk gave a really nice intro overview of how the Mahor subspecies were brought into captivity. But we also know that the Adra gazelle, the other subspecies, Nangerdama ruficollis, is also brought into captivity and is mostly managed in North American zoo and ranch populations, and so ex situ populations there. So these two species have very different population histories in terms of their introduction into captivity. A larger number of individuals of Adra gazelles were brought into North America back in the late 1960s. And so we would expect that, this population today would have retained more genetic diversity over time. Whereas by the time Mahor gazelles were brought into captivity, as Teresa and Sonia mentioned in their talk, they had basically been shrunk to a very small number and essentially had gone through a bottleneck. And so we would expect less diversity that had been, in a loss of diversity over time. And even though the population has recovered because of successful captive breeding efforts, the impact of those sort of small population sizes on genome-wide variation can linger for quite a long time. So we wanted to explore, these two scenarios with just some real data. And given that we're in this age of genomics now, we can sequence whole genomes of animals and plants. And so what we did is we generated a reference genome assembly using next generation sequencing methods from one Adra gazelle from the Smithsonian Conservation Biology Institute captive population. And we also then generated whole genome sequences from two Adras and two Mahor gazelles also found in North American captive populations. And the data from those four individuals were then mapped to the reference individual. And with that mapping, you can then identify the variants, what we call single nucleotide polymorphisms or SNPs in those individuals. Now with the reference individual, we also can annotate the genome, meaning that we can decipher the different components of the genome, genes and non-genes or repetitive regions. That's really important for understanding how that variation is spread across the genome. And then once we have all this data, we can then use this for a multitude of analyses, including examining genome-wide diversity and signals of inbreeding, deleterious variants. We can examine historical demography, and we can also investigate the differences between the subspecies, the Mahor and the Adra gazelle. And that's what I'm going to be focusing on here today. So from our studies, we have found that as the expectations that I mentioned before, is that the Adra gazelle has higher genetic diversity, actually about 2 times more diversity than the Mahor gazelle. And again, given the different population histories during the 20th century of these two subspecies, this is exactly what we'd expect. So we find higher genome-wide diversity in the Adra gazelle, and that's shown here by the variants that are mapped across the chromosomes. So the reference individual that we sequence is what's called a chromosome level assembly. So we can actually then characterize variation on specific chromosomes of the dama gazelle. We see less signals of inbreeding in the Adra gazelle and also lower amounts of damaging mutations, whereas we see the opposite in the Mahor gazelle. Now, the idea is that, well, there's still appreciable diversity in the Mahor gazelles, the 2 individuals that we've sequenced so far. But there have been proposals to suggest that, perhaps this species could, this subspecies could be rescued by breeding them with Adra gazelles. And so this was first, suggested based on some mitochondrial DNA work that Helen Sen and her colleagues published back in 2014. And the idea that subspecies, even though we see these phenotypic differences between Adra and Mahor gazelles are very apparent, we can also ask the question, well, how genetically different really are these species at the genome level? And, would this provide some information and guidance on how to, manage this species and these subspecies themselves? There's concerns about, mixing different evolutionary or subspecies entities. And, those are shown there on the red circle at the bottom. So loss of genetic uniqueness, potential outbreeding depression, that is loss of fitness by breeding two differently adapted, potentially adapted forms, loss of phenotypic diversity, and loss of taxonomic integrity. But the benefits also have to be considered, because you're improving the gene flow among populations that might have not had real gene flow barriers before the populations became decreased. That increases the genetic diversity, that allows us to solve some of the genetic problems, and lower diversity and higher deleterious variation in the Mahor gazelle. And for the species as a whole, this might help increase the long-term resilience. So on the basis of the mitochondrial work, a experimental population was set up in back in 2016 in Alien Zoo. And so doing reciprocal crosses just to see what the effects would be in terms of on phenotype and offspring viability. And so these are the results that were reported in the 2019 Dama Gazelle conservation strategy. And, all things look positive so far. So what we wanted to do is take our handful of genomes and just investigate this a little more. So given the availability of other gazelle genomes like the Grant's gazelle, we could do a comparison. So what we did first is we took our Dama gazelle genomes and an available Grant's gazelle genome published by another group, and we did what's called a sliding window analysis. That is, we just went across the genomes of these individuals and either 50,000 base pairs, 100,000 base pairs, or 200,000 base pairs, and just sort of summed up the differences in each of those windows, and we can just average that. And so when we do that, we see, when we compare a Grant's gazelle with either an Adra or Mahor gazelle, we see 5 times more difference than we see between the two subspecies of Dama gazelle. And so that translates into a very small, less than 1% difference across the total genome of the Adra gazelle and the Mahor gazelle. We also looked at mitochondrial genomes. And so given the 6 individuals that we analyzed here, one coming from a previously published Dama gazelle mitochondrial genome, we also find a very small number of differences among these individuals, less than 1% between these subspecies. And this is shown very nicely between this very short branch length separating the ruficollis versus the Mahor gazelles. Compare that to their sister species, the Summerings gazelle and the Grant's gazelles. There's much more differences between these. And then when you go out to a different genus like the red-fronted gazelle, Eudorcus, you find even a higher amount of difference. We can also then use our nuclear genome data to ask, well, what does the historical population look like among these different subspecies? And so they are also very similar. So the idea is that genomes contain variations, within each individual, and that information that can actually be used to reconstruct the population history in terms of effective population size for the last million years or so. And so what we see is that, the red on top is the Adra, the blue lines on the bottom are the Mahors. We see a very similar population size trajectory. After about 1 million years ago, both subspecies populations declined. And we can also then do a additional analysis called hybrid pairwise sequential Markovian coalescent analysis. It's just a fancy word for a type of analysis that we can estimate. For example, when we can ask the question, when was the last time, based on our genomes, these two subspecies last shared genes with each other? That is basically, when did gene flow enter or end? And so we find that is about 65,000 years ago. And when we do additional modeling on this, we find that gene flow could have ended as little as 40,000 years ago, with some signals even more recently. So within, between 40,000 years ago and 10,000 years. So this suggests that these subspecies did, share a very recent common ancestor with each other and are relatively similar to each other and have had very similar population histories, despite, their historical range distribution and large separation between the two subspecies. And then one of the concerns with breeding, different organisms, especially those that show different patterns of chromosome numbers, like the Mahor and the Adder gazelles do, is that perhaps this different chromosomal rearrangement can cause a decrease in fitness and genetic inviability. Well, we were fortunate because the center of the Mahor gazelle captive breeding program in North America was the San Diego Zoo, and every animal that was bred at the San Diego Zoo was karyotyped. That is, scientists collected and generated chromosome spreads of each of the animals that was born through the captive breeding program. And so I was able to collaborate with Marlis Hook, who runs the cytogenetics laboratory at the San Diego Zoo, and had this great data that had been assembled over the captive breeding history of the Mahor gazelle, plus some adder gazelles that were karyotyped as well. And what we see is actually 3 different chromosome number patterns in the dama gazelle. So individuals with 38 chromosomes, 39 chromosomes, and 40. And what we see is that there is no specific association between chromosome number and different subspecies. And what was nice is that the San Diego Zoo also had the captive breeding pedigree, and so we could map these chromosome numbers or cytotypes across the different individuals in the pedigree, and we find no association between subspecies or individuals with certain types of karyotypes breeding other types of karyotypes. So meaning that there's just no assortative mating based on chromosome number. So all in all, this tells us a lot, is that, whether we should go ahead and breed these animals, that's still, an open question, and I think worthy of further study. And this is definitely something, for example, that Helen Sen and Cara Dix at the Royal Zoological Society of Scotland are currently pursuing. So what we've done with our dama gazelle genomes is we generated a panel of SNPs, 10,000 SNPs distributed across the genome of the dama gazelle. And those SNPs can be then turned into a tool that allows you to genotype hundreds of dama gazelles at the same time. So this is what Cara and Helen are now pursuing, and we were able to help them develop this tool. And this larger number of individuals will definitely shed additional light on some of the findings that we've generated with our handful of genomes. So with that, I just thank every one of my colleagues and collaborators who worked on this project, and would be happy to take questions. Thank you.